This complete audiobook made by Shalu Moria.
Unit 2. Structural Organization in Plants and Animals. Chapter 5 Morphology of Flowering Plants. Chapter 6 Anatomy of Flowering Plants. Chapter 7 Structural Organization in Animals. The description of the diverse forms of life on Earth was made only by observation, through naked eyes or later through magnifying lenses and microscopes. This description is mainly of gross structural features, both external and internal. In addition, observable and perceivable living phenomena were also recorded as part of this description. Before experimental biology, or more specifically, physiology, was established as a part of biology, naturalists described only biology. Hence, biology remained as a natural history for a long time. The description, by itself, was amazing in terms of detail. While the initial reaction of a student could be boredom, one should keep in mind that the detailed description was utilized in the later-day reductionist biology, where living processes drew more attention from scientists than the description of life forms and their structure. Hence, this description became meaningful and helpful in framing research questions in physiology or evolutionary biology. In the following chapters of this unit, the structural organization of plants and animals, including the structural basis of physiological or behavioral phenomena, is described. For convenience, this description of morphological and anatomical features is presented separately for plants and animals. Catherine Esau, 1898 to 1997. Catherine Esau was born in Ukraine in 1898. She studied agriculture in Russia and Germany and received her doctorate in 1931 in United States. She reported in her early publications that the curly top virus spreads through a plant via the food-conducting or phloem tissue. Dr. Esau's Plant Anatomy, published in 1954, took a dynamic, developmental approach designed to enhance one's understanding of plant structure and an enormous impact worldwide, literally bringing about a revival of the discipline. The Anatomy of Seed Plants by Catherine Esau was published in 1960. It was referred to as Webster's of Plant Biology. It is encyclopedic. In 1957, she was elected to the National Academy of Sciences, becoming the sixth woman to receive that honor. In addition to this prestigious award, she received the National Medal of Science from President George Bush in 1989. When Catherine Esau died in the year 1997, Peter Raven, director of Anatomy and Morphology, Missouri Botanical Garden, remembered that she absolutely dominated the field of plant biology even at the age of 99. Chapter 5. Morphology of Flowering Plants. 5.1 The root. 5.2 The stem. 5.3 The leaf. 5.4 The inflorescence. 5.5 The flower. 5.6 The fruit. 5.7 The seed. 5.8 Semi-technical description of a typical flowering plant. 5.9 Description of some important families. The wide range in the structure of higher plants will never fail to fascinate us. Even though the angiosperms show such a large diversity in external structure or morphology, they are all characterized by presence of roots, stems, leaves, flowers, and fruits. In chapters 2 and 3, we talked about classification of plants based and at understanding any higher plant, or for that matter any living organism, we need to know standard technical terms and standard definitions. We also need to know about the possible variations in different parts, found as adaptations of the plants to their environment, for example, adaptations to various habitats, for protection, climbing, storage, etc. If you pull out any weed, you will see that all of them have roots, stems, and leaves. They may be bearing flowers and fruits. The underground part of the flowering plant is the root system, while the portion above the ground forms the shoot system, figure 5.1. 5.1 The root. In majority of the dicotyledonous plants, the direct elongation of the radicle leads to the formation of primary root, which grows inside the soil. It bears lateral roots of several orders that are referred to as secondary, tertiary, etc. roots. The primary roots and its branches constitute the taproot system, as seen in the mustard plant, figure 5.2a. In monocotyledonous plants, the primary root is short-lived and is replaced by a large number of roots. These roots originate from the base of the stem and constitute the fibrous root system, as seen in the wheat plant, figure 5.2b. In some plants, like grass, monstera, and the banyan tree, roots arise from parts of the plant other than the radicle and are called adventitious roots, figure 5.2c. The main functions of the root system are absorption of water and minerals from the soil, providing a proper anchorage to the plant parts, storing reserve food material, and synthesis of plant growth regulators. Figure 5.1 Parts of a flowering plant. Root system. Shoot system. Secondary root. Primary root. Bud. Node, internode. Leaf. Stem. Fruit. Flower. Figure 5.2 Different types of roots. A. Tap. B. Fibrous. C. Adventitious. Laterals. Main root. Fibrous roots. Adventitious roots. 5.1.1 Regions of the root. The root is covered at the apex by a thimble-like structure called the root cap. Figure 5.3. Figure 5.3 The regions of the root tip. Root hair. Root cap. Region of meristematic activity. Region of elongation. Region of maturation. It protects the tender apex of the root as it makes its way through the soil. A few millimeters above the root cap is the region of meristematic activity. The cells of this region are very small, thin-walled, and with dense protoplasm. They divide repeatedly. The cells proximal to this region undergo rapid elongation and enlargement and are responsible for the growth of the root in length. This region is called the region of elongation. The cells of the elongation zone gradually differentiate and mature. Hence, this zone, proximal to region of elongation, is called the region of maturation. From this region, some of the epidermal cells form very fine figure 5.3 the regions of the root tip and delicate, thread-like structures called root hairs. These root hairs absorb water and minerals from the soil. 5.2 The stem. What are the features that distinguish a stem from a root? The stem is the ascending part of the axis bearing branches, leaves, flowers, and fruits. It develops from the plumule of the embryo of a germinating seed. The stem bears nodes and internodes. The region of the stem where leaves are born are called nodes, while internodes are the portions between two nodes. The stem bears buds, which may be terminal or axillary. Stem is generally green when young and later often become woody and dark brown. The main function of the stem is spreading out branches bearing leaves, flowers, and fruits. It conducts water, minerals, and photosynthase. Some stems perform the function of storage of food, support, protection, and of vegetative propagation. 5.3 The leaf. The leaf is a lateral, generally flattened structure borne on the stem. It develops at the node and bears a bud in its axil. The axillary bud later develops into a branch. Leaves originate from shoot apical meristems and are arranged in an acropoidal order. They are the most important vegetative organs for photosynthesis. A typical leaf consists of three main parts: leaf base, petiole, and lamina, figure 5.4a. Figure 5.4 Structure of a leaf. A. Parts of a leaf. Lamina. Stipule. Petiole. Leaf base. Axillary bud. The leaf is attached to the stem by the leaf base and may bear two lateral small leaf-like structures called stipules. In monocotyledons, the leaf base expands into a sheath covering the stem partially or wholly. In some leguminous plants, the leaf base may become swollen, which is called the pulvinus. The petiole help hold the blade to light. Long, thin, flexible petioles allow leaf blades to flutter in wind, thereby cooling the leaf and bringing fresh air to leaf surface. The lamina or the leaf blade is the green expanded part of the leaf with veins and veinlets. There is, usually, a middle prominent vein, which is known as the midrib. Veins provide rigidity to the leaf blade and act as channels of transport for water, minerals, and food materials. The shape, margin, apex, surface, and extent of incision of lamina varies in different leaves. 5.3.1 Venation. The arrangement of veins and the veinlets in the lamina of leaf is termed as venation. When the veinlets form a network, the venation is termed as reticulate, figure 5.4b. Figure 5.4 Structure of a leaf, B. Reticulate venation. When the veins run parallel to each other within a lamina, the venation is termed as parallel, figure 5.4c. Figure 5.4 Structure of a leaf, C. Parallel venation. Leaves of dicotyledonous plants generally possess reticulate venation, while parallel venation is the characteristic of most monocotyledons. 5.3.2 Types of leaves. A leaf is said to be simple when its lamina is entire or when incised, the incisions do not touch the midrib. When the incisions of the lamina reach up to the midrib, breaking it into a number of leaflets, the leaf is called compound. A bud is present in the axil of petiole in both simple and compound leaves, but not in the axil of leaflets of the compound leaf. The compound leaves may be of two types, figure 5.5. Figure 5.5 Compound leaves, A. Pinnately compound leaf, B. Palmately compound leaf. A. Neem. B. Silk cotton. Rachis. In a pinnately compound leaf, A. Number of leaflets are present on a common axis, the rachis, which represents the midrib of the leaf as in neem. In palmately compound leaves, the leaflets are attached at a common point, that is, at the tip of petiole, as in silk cotton. 5.3.3 Phyllotaxy. Phyllotaxy is the pattern of arrangement of leaves on the stem or branch. This is usually of three types, alternate, opposite, and whorled, figure 5.6. Figure 5.6 Different types of phyllotaxy. A. Alternate. B. Opposite. C. Whorled. A. China rose. B. Guava. C. Alstonia. In alternate type of phyllotaxy, a single leaf arises at each node in alternate manner, as in China rose, mustard, and sunflower plants. In opposite type, a pair of leaves arise at each node and lie opposite to each other, as in Calotropis and guava plants. If more than two leaves arise at a node and form a whorl, it is called whorled, as in Alstonia. 5.4 The inflorescence. A flower is a modified shoot wherein the shoot apical meristem changes to floral meristem. Internodes do not elongate and the axis gets condensed. The apex produces different kinds of floral appendages laterally at successive nodes instead of leaves. When a shoot tip transforms into a flower, it is always solitary. The arrangement of flowers on the floral axis is termed as inflorescence. Depending on whether the apex gets developed into a flower or continues to grow, two major types of inflorescences are defined, racemose and cymose. In racemose type of inflorescences, the main axis continues to grow. The flowers are borne laterally in an acropoidal succession, figure 5.7. In cymose type of inflorescence, the main axis terminates in a flower, hence is limited in growth. The flowers are borne in a basipital order, figure 5.7. Figure 5.7 Racemose inflorescence. 5.5 The flower. The flower is the reproductive unit in the angiosperms. It is meant for sexual reproduction. A typical flower has four different kinds of whorls arranged successively on the swollen end of the stalk or pedicel, called thalamus or receptacle. These are calyx, corolla, andrecium, and gynoecium. Calyx and corolla are accessory organs, while andrecium and gynoecium are reproductive organs. In some flowers like lily, the calyx and corolla are not distinct and are termed as perianth. When a flower has both androecium and gynoecium, it is bisexual. A flower having either only stamens or only carpels is unisexual. In symmetry, the flower may be actinomorphic, radial symmetry, or zygomorphic, bilateral symmetry. When a flower can be divided into two equal radial halves in any radial plane passing through the center, it is said to be actinomorphic, for example, mustard, datura, chili. When it can be divided into two similar halves only in one particular vertical plane, it is zygomorphic, for example, pea, gulmour, bean, cassia. A flower is asymmetric, irregular, if it cannot be divided into two similar halves by any vertical plane passing through the center, as in canna. A flower may be trimerous, tetramerous, or pentamerous when the floral appendages are in multiple of 3, 4, or 5, respectively. Flowers with bracts-reduced leaf found at the base of the pedicel are called bracteate, and those without bracts, ebracteate. Figure 5.8 Cymose inflorescence. Figure 5.9 Position of floral parts on thalamus, A. Hypogynous. B. and C. Perigynous. D. Epigynous. Based on the position of calyx, corolla, and androecium in respect of the ovary on thalamus, the flowers are described as hypogynous, perigynous, and epigynous, figure 5.9. In the hypogynous flower, the gynoecium occupies the highest position while the other parts are situated below it. The ovary in such flowers is said to be superior, for example, mustard, China rose, and brinjal. If gynoecium is situated in the center and other parts of the flower are located on the rim of the thalamus almost at the same level, it is called perigynous. The ovary here is said to be half inferior, for example, plum, rose, peach. In epigynous flowers, the margin of thalamus grows upward enclosing the ovary completely and getting fused with it. The other parts of flower arise above the ovary. Hence, the ovary is said to be inferior as in flowers of guava and cucumber, and the ray florets of sunflower. 5.5.1 Parts of a flower. Each flower normally has 4 floral whorls, viz., calyx, corolla, androecium, and gynoecium, figure 5.10. Figure 5.10 Parts of a flower. Androecium, gynoecium, corolla, calyx, pedicel, calyx, corolla, androecium, gynoecium. 5.5.1.1 Calyx. The calyx is the outermost whorl of the flower and the members are called sepals. Generally, sepals are green, leaf-like, and protect the flower in the bud stage. The calyx may be gamosepalous, sepals united, or polysepalous, sepals free. 5.5.1.2 Corolla. Corolla is composed of petals. Petals are usually brightly colored to attract insects for pollination. Like calyx, corolla may also be gamopetalous, petals united, or polypetalous, petals free. The shape and color of corolla vary greatly in plants. Corolla may be tubular, bell-shaped, funnel-shaped, or wheel-shaped. Estivation, the mode of arrangement of sepals or petals in floral bud with respect to the other members of the same whorl, is known as estivation. The main types of estivation are valvate, twisted, imbricate, and vexillary, figure 5.11. Figure 5.11 Types of estivation in corolla: A. Valvate, B. Twisted, C. Imbricate, D. Vexillary. When sepals or petals in a whorl just touch one another at the margin, without overlapping, as in Calotropis, it is said to be valvate. If one margin of the appendage overlaps that of the next one and so on as in China rose, lady's finger, and cotton, it is called twisted. If the margins of sepals or petals overlap one another but not in any particular direction as in cassia and gulmour, the estivation is called imbricate. In pea and bean flowers, there are 5 petals. The largest, standard, overlaps the 2 lateral petals, wings, which in turn overlap the 2 smallest anterior petals, keel. This type of estivation is known as vexillary or papilionaceous. 5.5.1.3 Andrecium. Andrecium is composed of stamens. Each stamen, which represents the male reproductive organ, consists of a stalk or a filament and an anther. Each anther is usually bilobed, and each lobe has 2 chambers, the pollen sacs. The pollen grains are produced in pollen sacs. A sterile stamen is called staminode. Stamens of flower may be united with other members such as petals or among themselves. When stamens are attached to the petals, they are epipetalous as in brinjal, or epiphyllous when attached to the perianth as in the flowers ophyllae. The stamens in a flower may either remain free, polyandrous, or may be united in varying degrees. The stamens may be united into one bunch or one bundle, monoadelphous, as in China rose, or 2 bundles, diadelphous, as in pea, or into more than 2 bundles, polyadelphous, as in citrus. There may be a variation in the length of filaments within a flower, as in salvia and mustard. 5.5.1.4 Gynoecium. Gynoecium is the female reproductive part of the flower and is made up of 1 or more carpels. A carpel consists of 3 parts, namely stigma, style, and ovary. Ovary is the enlarged basal part, on which lies the elongated tube, the style. The style connects the ovary to the stigma. The stigma is usually at the tip of the style and is the receptive surface for pollen grains. Each ovary bears 1 or more ovules attached to a flattened, cushion-like placenta. When more than 1 carpel is present, they may be free, as in lotus and rose, and are called apicarpous. They are termed syncarpous when carpels are fused, as in mustard and tomato. After fertilization, the ovules develop into seeds and the ovary matures into a fruit. Placentation, the arrangement of ovules within the ovary, is known as placentation. The placentation are of different types, namely marginal, axial, parietal, basal, central and free central, figure 5.12. Figure 5.12 Types of placentation. I. Marginal. B. Axial. C. Parietal. D. Free central. E. Bassa. In marginal placentation, the placenta forms a ridge along the ventral suture of the ovary, and the ovules are borne on this ridge, forming 2 rows, as in P. When the placenta is axial and the ovules are attached to it in a multilocular ovary, the placenta ion is said to be axial, as in China rose, tomato, and lemon. In parietal placentation, the ovules develop on the inner wall of the ovary or on peripheral part. Ovary is 1-chambered, but it becomes 2-chambered due to the formation of the false septum, for example, mustard and argemone. When the ovules are borne on central axis and septa are absent, as in dianthus and primrose, the placentation is called free central. In basal placentation, the placenta develops at the base of ovary and a single ovule is attached to it, as in sunflower, marigold. 5.6 The fruit. The fruit is a characteristic feature of the flowering plants. It is a mature or ripened ovary, developed after fertilization. If a fruit is formed without fertilization of the ovary, it is called a parthenocarpic fruit. Generally, the fruit consists of a wall or pericarp and seeds. The pericarp may be dry or fleshy. When pericarp is thick and fleshy, it is differentiated into the outer epicarp, the middle mesocarp, and the inner endocarp. In mango and coconut, the fruit is known as a drupe, figure 5.13. They develop from monocarpellary superior ovaries and are 1-seeded. In mango, the pericarp is well differentiated into an. Figure 5.13 Parts of a fruit. A. Mango. B. Coconut. Epicarp. Mesocarp. Seed. Endocarp. Outer thin epicarp, a middle fleshy edible mesocarp and an inner stony hard endocarp. In coconut, which is also a drupe, the mesocarp is fibrous. 5.7 The seed. The ovules after fertilization develop into seeds. A seed is made up of a seed coat and an embryo. The embryo is made up of a radicle, an embryonal axis and one, as in wheat, maize, or two cotyledons, as in gram and pea. 5.7.1 Structure of a dicotyledonous seed. The outermost covering of a seed is the seed coat. The seed coat has two layers, the outer testa and the inner tegmen. The hilum is a scar on the seed coat through which the developing seeds were attached to the fruit. Above the hilum is a small pore called the micropyle. Within the seed coat is the embryo, consisting of an embryonal axis and two cotyledons. The cotyledons are often fleshy and full of reserved food materials. At the two ends of the embryonal axis are present the radicle and the plumule, figure 5.14. Figure 5.14 Structure of dicotyledonous seed. Seed coat. Micropyle. Hilum. Cotyledon. Plumule. Radicle. In some seeds, such as castor, the endosperm formed as a result of double fertilization is a food-storing tissue and called endospermic seeds. In plants such as bean, gram, and pea, the endosperm is not present in mature seeds, and such seeds are called non-endospermous. 5.7.2 Structure of monocotyledonous seed. Generally, monocotyledonous seeds are endospermic, but some, as in orchids, are non-endospermic. In the seeds of cereals such as maize, the seed coat is membranous and generally fused with the fruit wall. The endosperm is bulky and stores food. The outer covering of endosperm separates the embryo by a proteinous layer called alurone layer. The embryo is small and situated in a groove at one end of the endosperm. It consists of one large and shield-shaped cotyledon known as scutellum and a short axis with a plumule and a radicle. The plumule and radicle are enclosed in sheaths which are called coleoptile and coleorhiza respectively, figure 5.15. Figure 5.15 Structure of a monocotyledonous seed. Embryo. Endosperm. Alurone layer. Seed coat and fruit wall. Endosperm. Scutellum. Coleoptile. Plumule. Radicle. Coleorhiza. 5.8 Semi-technical description of a typical flowering plant. Various morphological features are used to describe a flowering plant. The description has to be brief, in a simple and scientific language, and presented in a proper sequence. The plant is described beginning with its habit, vegetative characters, roots, stem and leaves, and then floral characters, inflorescence, and flower parts. After describing various parts of plant, a floral diagram and a floral formula are presented. The floral formula is represented by some symbols. In the floral formula, BR stands for bractea, K stands for calyx, C for corolla, P for perianth, F for androecium, and G for gynoecium, G for superior ovary and G for inferior ovary, for male, for female, for bisexual plants, circle plus 4 actinomorphic. Figure 5.16 Floral diagram with floral formula. N% 4 zygomorphic nature of flower. Fusion is indicated by enclosing the figure within bracket and adhesion by a line drawn above the symbols of the floral parts. A floral diagram provides information about the number of parts of a flower, their arrangement, and the relation they have with one another, figure 5.16. The position of the mother axis with respect to the flower is represented by a dot on the top of the floral diagram. Calyx, corolla, androecium, and gynoecium are drawn in successive whorls, calyx being the outermost and the gynoecium being in the center. Floral formula also shows cohesion and adhesion within parts of whorls and between whorls. The floral diagram and floral formula in figure 5.16 represents the mustard plant, family, Brassicaceae. 5.9 Solanaceae. It is a large family, commonly called as the potato family. It is widely distributed in tropics, subtropics, and even temperate zones. Figure 5.17. Figure 5.17 Solanum nigrum, Makoi, plant, A, flowering twig, B, flower, C, ls of flower, D, stamens, E, carpel, F, floral diagram. Vegetative characters: plants mostly herbs, shrubs, and rarely small tree stem. Herbaceous, rarely woody, aerial, erect, cylindrical, branched, solid or hollow, hairy or glabrous, underground stem in potato, Solanum tuberosum. Leaves: alternate, simple, rarely pinnately compound, exstipulate, venation reticulate. Floral characters: inflorescence, solitary, axillary or cymose as in Solanum. Flower: bisexual, actinomorphic. Calyx: sepals 5, united, persistent, valvate estivation. Corolla: petals 5, united, valvate estivation. Andrecium: stamens 5, epipetalous. Gynoecium: bicarpellary, obligately placed, syncarpous, ovary superior, bilocular, placenta swollen with many ovules, axile. Fruits: berry or capsule. Seeds: many, endospermous. Floral formula: circle plus. Economic importance: many plants belonging to this family are source of food: tomato, brinjal, potato, spice, chili, medicine, belladonna, ashwagandha, fumigatory, tobacco, ornamentals, petunia. Summary: flowering plants exhibit enormous variation in shape, size, structure, mode of nutrition, lifespan, habit, and habitat. They have well-developed root and shoot systems. Root system is either taproot or fibrous. Generally, dicotyledonous plants have taproots while monocotyledonous plants have fibrous roots. The roots in some plants get modified for storage of food, mechanical support, and respiration. The shoot system is differentiated into stem, leaves, flowers, and fruits. The morphological features of stems, like the presence of nodes and internodes, multicellular hair, and positively phototropic nature, help to differentiate the stems from roots. Leaf is a lateral outgrowth of stem developed exogenously at the node. These are green in color to perform the function of photosynthesis. Leaves exhibit marked variations in their shape, size, margin, apex, and extent of incisions of leaf blade, lamina. The flower is a modified shoot, meant for sexual reproduction. The flowers are arranged in different types of inflorescences. They exhibit enormous variation in structure, symmetry, position of ovary in relation to other parts, arrangement of petals, sepals, ovules, etc. After fertilization, the ovary is modified into fruits and ovules into seeds. Seeds either may be monocotyledonous or dicotyledonous. They vary in shape, size, and period of viability. The floral characteristics form the basis of classification and identification of flowering plants. This can be illustrated through semi-technical descriptions of families. Hence, a flowering plant is described in a definite sequence by using scientific terms. The floral features are represented in the summarized form as floral diagrams and floral formula. Exercises. 1. How is a pinnately compound leaf different from a palmately compound leaf? 2. Explain with suitable examples the different types of phyllotaxy. 3. Define the following terms. A. Estivation. B. Placentation. C. Actinomorphic. D. Zygomorphic. E. Superior ovary. F. Perigynous flower. G. Epipetalous stamen. 4. Differentiate between A. Racemose and cymose inflorescence. B. Apocarpous and syncarpous ovary. 5. Draw the label diagram of the following. I. Gram seed. 2. VS of maize seed. 6. Take one flower of the family Solanaceae and write its semi-technical description. Also draw their floral diagram. 7. Describe the various types of placentations found in flowering plants. 8. What is a flower? Describe the parts of a typical angiosperm flower. 9. Define the term inflorescence. Explain the basis for the different types. Inflorescence in flowering plants. 10. Describe the arrangement of floral members in relation to their insertion on thalamus.